bass. The name of our project is Culture Litter-Induced Intersex in Regional Largemouth Bass Population. So to start our presentation, we want to introduce you to someone very special to our team. He's our team mascot, and his name is Bob. Bob is an 18-month-old largemouth bass. Largemouth bass are native to the Chesapeake Bay and also significant keystone predators. They function to maintain organization and diversity in the ecological community. Because of this, any significant change in their population levels can have wide-ranging effects on the rest of the aquatic ecosystem. That's why it was especially concerning when in 2002, massive fish kills were found throughout the Chesapeake Bay and the Shenandoah River Valley. The majority of the fish killed were smallmouth bass, a close relative of the largemouth bass. Similar mortality events like this occurred in 2004, 2005, and 2009, so this wasn't an isolated incident. What was most shocking about this discovery was that when scientists opened up these fish and histologically examined their gonads, they found the presence of testicular oocytes, which are female germ cells which had grown in the testicles of these male fish. This is obviously abnormal, and they termed this feminization of the male fish intersex. And scientists are currently investigating whether intersex is affecting the reproductive system of these fish as well as the immune functioning, which could potentially lead to their death and the multiple fish kills. So that leads us to one of the big questions of our research, which is what is causing the intersex in these fish? We believe that it is a symptom of a wider global pollution problem, and I'm going to let Shimani talk to us more about this.
So initial research on this phenomenon of intersex showed an interesting trend. Scientists were looking, and they found that the occurrence of intersex was usually downstream of urban areas as well as industrial sources of pollution, such as wastewater treatment plants and pulp and paper mills. Now, these sources emit sewage effluent into nearby bodies of water. And in the sewage effluent, scientists were discovering what are known as endocrine disrupting compounds, or EDCs for short. Now, we know that EDCs enter the body and compromise normal hormone function, which can ultimately lead to abnormal conditions such as intersex, or in severe cases, massive fish kills, as Taylor mentioned earlier. What's interesting about Maryland is that while we do have these aforementioned sources of EDC occurring in the state, we also have along the eastern shore occurrences of intersex, but not because of the same sources, because the eastern shore is an agriculturally intensive region consisting mostly of farmland and little to no industrial activity. So, as you can see on the map here, the dark red represents industrial activity, and as you can see on the right side, there's only light green, which is for agriculture. So one potential source of EDCs in this region then is agricultural runoff of crop fertilizer from farms, which enters the water during heavy rainfall. Such fertilizer consists of poultry litter or chicken fecal matter, which is collected, dried, and mixed with bedding as well as other nutrients that nourish farm crops. Now to farmers, this is a lot more beneficial to their farms because it's cheaper and more natural, but all organisms excrete a natural amount of hormones in their waste, and because poultry litter is chicken food, it is no exception. Therefore, we can suggest that poultry litter is entering the Chesapeake Bay tributaries as a potential source of these EDCs and ultimately causing intersex in this region. That leads us to our two research questions. So now that we've noticed a relationship between poultry litter and intersex, we want to see if we can induce intersex in a laboratory setting using poultry litter, specifically with largemouth bass populations. We're also interested in seeing if age plays any role in this, whether the fish are more sensitive to the EDCs at a younger age, or whether the time of exposure affects the severity of intersex development later on in life. So to answer our first question, we've exposed a total of 525 fish across 3 different treatments. We first have our negative control of just plain water, in which we expect to see no change and no development of any type of condition. We also have our positive control of 17-beta estradiol, which is an estrogen and known EDC. In these fish, we do expect to see intersex development because this estrogen is very potent. Finally, we have our poultry litter treatment, in which we don't know what to fully expect, but we can say that we will see some level of endocrine disruption, if not full-on intersex. To answer our second question, we've stratified these 525 fish across different ages. So as you can see on the slide, we have 4 different ages that we're interested in looking at. Over the summer, we successfully completed our exposures to our 3-month-old and 12-month-old bass, and we're currently in the process of exposing our 5-month-old and 18-month-old bass. We're actually planning on finishing that this coming Saturday, so we're excited to get into the laboratory and work just all day. And just to give you an idea of what our exposures look like, so this is our mentor's lab in animal sciences, and we have 3 different takes for the 3 different treatments. There's culture litter in the back, estradiol in the middle, and our negative control water in the front. I'll now hand it off to Winston, who will talk a little bit more about our exposures in detail, as well as data collection and analysis.
Thanks, Shivani. Now we get to talk about the really exciting stuff, so I hope you guys pay attention, all right? So, as Shivani mentioned, we're doing exposures of fish over time. So if you consider day 0, the day when we bring our fish directly to the facility, we expose them for about 2 weeks. So they finish their exposure on day 14. After day 14, we take them out of that exposure and we let them grow out until they're approximately 2 years, until they're more sexually mature. And so then we can look and see if there's development of intersex and the like. Now, at the end of our exposure, and when they are 2 years old, we unfortunately have to sacrifice some of our fish. We typically sacrifice until we get about 8 males because we really want to see the feminization in the males. So we normally sacrificing around 15 fish from each of our subsets. And from those sacrificed fish subsets, we then run different analyses to look for the presence of intersex. And this kind of analysis really are in 2 forms for us. Our first form is looking at histological sections. So just like what Taylor showed you earlier, we take the gonad of the fish out of the fish, and then we slice it up into thin sections. And then we look to see if there's the presence of these testicular oocytes. And that's a really well documented, it's kind of the gold standard for intersex detection. And we're also doing something that's really cool. We're looking for the presence of a protein, which is normally only made by females. And so this protein is called vitellogenin, it's an egg protein. And so if a female protein is found in a male fish, that's kind of a problem. And so that's how we're going to define someone as also having intersex. And so we'll be using an ELISA assay, which pretty much is just an assay that binds specifically to the protein and will give off a certain fluorescent wavelength of light so that we can, when light is shined upon it, so that we can measure and quantify the level of intersex. All right, so the second thing we wanted to look at, other than whether these fish were changing into these kind of intersex type characteristics, was to see what in the poultry litter, what in that stuff was actually causing this to happen. And so to do this, we had to do some water chemistry. So all along the way, all through our 14-day exposure, we've been collecting water samples and just kind of freezing them up. And from that, we're going to run different analyses to see what's exactly in that water, what's causing the intersex. And so there's 2 things we're doing here as well. The first thing is we're trying to look and see specifically what chemicals, what estrogens are in the water. And we're really excited about this. We are partnering with the State University of New York at Buffalo. Dr. Diana Aga, she's an expert in the field. She's been doing this for like 40 years, which is really, really great. And we actually just went up to Buffalo a couple weeks ago, and we brought a bunch of our samples, and she's running them right now. So we're really excited to see what's going to happen. She has a liquid chromatography dual mass spec machine. Big mouthful, but really all it is just measuring and quantifying the levels and detecting different estrogens which are in our water samples. Second thing we're going to do is look at estrogenicity. And estrogenicity is basically not the particular molecule, but looking at the activity of these molecules. So we're going to use something called a yeast assay, and this yeast is very cool because if the estrogen starts reacting with the yeast will light up. So it's actually a bioluminescent assay. And using that, we can quantify to see whether or not there's estrogen activity in the water. So, to summarize, the oocytes in these fish that we have been finding are a presence of some weird and wacky phenomena going on, and we think that this could be symptomatic of a larger global issue related to endocrine disruption. And so we specifically want to see whether poultry litter, which is an agricultural contaminant, whether that can cause intersex. And we also want to understand at what ages these fish are most sensitive. We want to see whether it's baby fish when they're first exposed, when they're born, or whether these older fish that have been swimming around in our lakes and rivers for a long time, whether when there is effluent from there, that they can also develop this intersex. And so to that end, we've exposed over 500 fish to things, and as she mentioned, we're going to be finishing our next exposure on Saturday, so it'll be 500 fish. And we also have collected plasma samples for that egg protein analysis. We've also collected histological samples for our analysis for looking for testicular oocytes. And we've also collected samples for water analysis and chemistry. And in the future, we still intend to do more exposures. Hopefully in the spring, we're going to do a big exposure. And we also want to analyze all of this vast data we've collected. So we haven't really siphoned through all this data yet, so we're not going to present that to you yet. But hopefully you'll see it at thesis conference. It'll be exciting. And of course, nothing is very simple in Gemstone, and so we've had a lot of challenges. Probably our biggest challenge, right in the middle, we tried to do an exposure of baby fish, just hatched fish, less than a year old, 1,000 of these fish. We were super excited. Every single fish died. So that whole sample was completely gone. We've readjusted the water chemistry in our system, so we're hoping to re-expose them in May of next year, so cross our fingers, hope that goes well. Another big issue is with faulty technology. A lot of our tests are very, very, very specific, and so the instrumentation is really, really fickle. And so if you mess something up and it goes broke, you have to find a way to buy a new one. And so that really ties me into another piece of advice we want to give to freshmen. You really, really, really need to start looking for grants early, finding money for your Gemstone teams. We've been blessed. We've been receiving a grant from the Maryland Water Resources Commission, and we've also gotten grants from the National NOAA Sea Grant and the ACC-IAC grant, so we've been doing okay on that. The other thing we found are these parasites that have been growing in our fish. When we first found them, we were freaking out because we thought it might mess with the other fish in our system, so we had to find a way to quarantine those fish off to prevent that from messing with their system, and we've given them the right antibiotics and treatment to fix that, and so hopefully that won't be an issue. And further advice for our freshmen, we can't stress this enough, but the most important person on your team is your mentor. So please go out there and find a good mentor. Find someone that's really going to promote you, find someone that's really going to care about you, and put in the effort to help your team succeed. Without your mentor, you're nothing, okay? The other thing, second thing, is to organize everything. You will not believe how many hours we have sat in our meetings just trying to figure out what we did those first 5 weeks of the year when we didn't write everything down. So if you just start from the very beginning, label everything, write everything, you will be much better off in the end. Last thing on our list is to balance work with fun. We do a lot of work in the lab all the time, but we never forget to have fun. Gemstone is a long process. You're going to stay with the same, we only have 8 people for 3 years. You really want them to be your friends, so we have family dinners and stuff, so do that. It's a lot of fun. It's great. We just want to thank Dr. Yankros, obviously, Dr. Aga up at SUNY Buffalo, and of course the Gemstone staff for being so supportive of us throughout this entire process. And we'd like to open the floor to any questions. Thank you.